Welcome to the Nexstim Podium webinar series. My name is Kate Lapp. I work for Nexstim and I will be helping to orchestrate today's webinar. So just a few words while we give people a couple more minutes to sign in. First, I would like to thank everyone for joining. We are very excited to see so many of you from all over the world with us today. And I'd like to extend a sincere thank you to Dr. Tep Casarotto for being here with us to present on the topic of how to collect and distinguish genuine EEG responses to NTMS. Now, for all of you who are joining us for the first time, welcome. We are very glad to have you here with us. And just a little background for you, the Nexstim Podium webinar series is meant to help share practical knowledge as well as the latest advances from our users all over the world so that we can learn from each other's experiences and advance the field together. These webinars will be available on demand later, and we'll send you a link to the recording in a few days in case you would like to share it with your colleagues. Now, today's webinar is on TMS evoked EEG, and this is a very hot topic right now. We had such an overwhelming response to a TMS EEG webinar a few weeks ago given by Dr. Marcello Massimini that we decided to hold a second follow-up webinar today, more practical in nature, so that those interested in this line of research have a better understanding on how to set up TMS EEG configurations, how to do the signal processing, and manage artifacts. Additionally, one aspect of Dr. Casarotto's work that I personally find very impressive is being able to do all of the signal processing and manage artifacts in real time. It can be a real challenge for any neurophysiology study, and Dr. Casarotto's team has a particularly nice approach. So we are very excited she's here with us today to share her expertise with you. Today's webinar will take about 50 to 60 minutes, and that does include the Q&A session at the end. The webinar is being recorded. Everybody except for Dr. Casarotto and myself are permanently muted, and your cameras are all inactive, so just sit back, relax. You don't need to worry about being visible. But because you are all muted, questions will be asked in the Q&A chat box, and you can find that by the icon with the question mark. You can send questions during and after the talk. However, they will only be responded to in the Q&A session at the end. Questions are anonymous unless you provide that information. And until your question is being answered in the Q&A session, it will only be visible for the organizers. Now, it's important to disclose that while Nexstim is hosting this webinar series, Nexstim does not have any influence over the content of the speaker's presentation. We do have FDA clearances for specific indications of use, namely pre-procedural language and motor mapping and RTMS treatment for major depressive disorder. We want to disclose that today's talk involves the use of navigated TMS that is currently not FDA cleared. This is for investigational use only at this time. And now I'm honored to introduce to you Dr. Tep Casarotto. Dr. Casarotto is an assistant professor of human physiology at the University of Milan. She received a PhD in bioengineering from the Polytechnic University of Milan and followed with a postdoc at the University of Pisa. She specializes in the advanced signal processing and data analysis of neurophysiological data. Dr. Casarotto has a unique expertise in the integration of functional imaging with clinical neurophysiology data, including fMRI and EEG. And she is now working to advance the field of TMS EEG within the group led by Professor Marcello Massimini. Her team has a special focus in developing and implementing various objective indices derived from TMS EEG data. Some of these indices have helped track brain plasticity in response to treatment, and others have been used to assess the potential for behaviorally unresponsive patients to regain consciousness. And because of the successes in her team's methods and advances, Dr. Casarotto has taken a large role in facilitating and standardizing the use of this technique across laboratories worldwide. and also standardizing the technique across different types of equipment. So Dr. Casarotto, we thank you for all of your contributions and advancing the field, and especially your efforts in educating the community about TMS EEG. It is such an honor to have you here with us today. Now, if you give me one moment, I will hand it over to you. Okay, your camera is up. The podium is yours.
Thank you very much. Good afternoon, everybody. Thank you, Katie, for this very nice introduction. I'm really happy to be here and to contribute to this podium series organized by Nexting. And I'm happy to be here today because my talk is rather methodological, as you already mentioned. And I think that it is very important to share experience and well-tested procedures across labs, because this approach, in my view, may really promote the development of good science in any field. So I can start sharing my screen. Okay. My talk is entitled How to Collect and Distinguish Genuine EEG Responses to Navigated TMS. The combination of transcranial magnetic stimulation and electroencephalography is very useful to measure cortical excitability and effective connectivity. With TMS, we can directly and non-invasively stimulate cortical neurons. With EEG, we can measure the overall brain's reaction to a focal perturbation. But when selecting TMS parameters, we have a full freedom of choice, because we have to decide where to stimulate, at what intensity, and with which 3D rotation of the column. In addition, there are other factors that cannot be well controlled a priori, but may affect the actual impact of TMS on the cortex. And for example, these factors include neuronal excitability, axonal displacement, and mesoscale anatomy in the stimulated site. So we may say that it is difficult to predict to what extent a certain combination of TMS parameters will actually activate cortical neurons. But let's start from what we know. We know that a direct and effective stimulation of the cortex should produce a local activation that progressively spreads towards distant sites, according to functional as well as anatomical connections. So in principle, if we were able to monitor EEG responses to TMS in real time, we could play with TMS parameters in order to improve the signal-to-noise ratio. In other words, we could adjust TMS parameters based on a real-time EEG readout already during the experiment in order to minimize the contribution of artifacts and at the same time maximize the impact of TMS on the cortex. This approach sounds fancy, but it's not novel at all, because, for example, it is routinely applied during ultrasound investigation. In this case, before evaluating a target organ, the operator empirically adjusts the precise location and orientation of the ultrasound beam in order to get a robust echo. But this approach is also not novel even within the TMS field, because it is commonly applied, for example, when collecting peripheral motor evoked potentials. In this case, the TMS operator arbitrarily sets stimulation intensity and coarsely places the coil over the primary motor cortex, often without relying on a neuronavigation system, but simply on scalp anatomical landmarks, and then starts stimulating. While delivering the first pulses, the operator empirically adjusts the precise location and orientation of the coil and the stimulation intensity until the electromyographic activity recorded from a target muscle satisfies specific amplitude and latency requirements. So why shouldn't we apply this same approach also when running TMS EEG experiments? I think it is perfectly reasonable to allow a TMS EEG operator to empirically adjust TMS parameters based on a real-time EEG readout. Clearly, recording EEG responses to TMS is technically more challenging compared to recording motor-evoked potentials, for two main reasons. First, motor-evoked potentials can be easily recorded by placing a single bipolar derivation over a muscle, whereas EEG responses to TMS are commonly recorded from several tens of EEG electrodes distributed on the scalp. Second, EEG signal is typically lower in amplitude as compared to EMG signal, and thus it can be more easily contaminated by artifacts. So in practice, we need a dedicated software tool that provides a customized display of TMS EEG data in real time. And in particular, we want this software tool to be able to show single trial EEG responses to TMS in real time. This is very important because large artifacts with a distinctive time course can be easily detected on single trial data. In addition, we want this software tool to be able to show also average TMS evoked potentials in real time. And this is also very important because from a provisional average of the first trials, for example, the first 20 trials, a TMS EEG operator can already measure coarsely but reliably the signal-to-noise ratio of TMS evoked potentials, for example, by comparing the amplitude of post-stimulus components with baseline EEG. In addition, using this provisional average, the operator can also check the possible occurrence of spurious components, which might be lower in amplitude, but also time-locked to TMS, and that's why they may arise only after averaging a few trials. This is, for example, the case of auditory potentials evoked by the coil's click, which may superimpose to genuine EEG responses to TMS if proper masking procedures are not applied during TMS EEG experiments. Now, is this software tool available? Well, the TMS-compatible EEG amplifiers currently on the market, unfortunately, are not equipped with such software. But fortunately, about 20 years ago, the Nexim company developed a TMS-compatible EEG amplifier specifically designed to collect TMS EEG data. Although this EEG amplifier is not available anymore, in our lab, inspired by this older system, we have developed a graphical user interface that is compatible with different EEG amplifiers currently on the market. This software tool works within the MATLAB environment, and the software code will be made freely available pretty soon. I want to highlight here that this software tool is just for customized display of TMS EEG data in real time. This software tool does not save anything on the hard disk because actual data collection and recording is in charge of the pre-proprietary softwares of the different EEG amplifiers. In the rest of my talk, I want to show you now how the perspective of a TMS EEG operator may change when using this software for real TMS EEG experiments. So imagine that we are about to start a new TMS EEG measurement session. So we can arbitrarily set TMS intensity, and we can place the coil somewhere on the scalp with a certain orientation. We deliver a single pulse and we ask, what do we see on the screen in real time? Barely nothing, because of the pulse artifact. This artifact spreads everywhere across all channels and is huge. It's 3 orders of magnitude larger than brain activity. So there's no way to set the amplitude range of the signal in order to see both the artifact and EEG. The pulse artifact is produced by the interaction between the strong and impulsive magnetic field produced by TMS on one hand and the EEG leads and amplifiers circuitry on the other. Fortunately, the duration of this artifact exclusively depends on 2 factors: electrode shape and hardware characteristics of the EEG amplifier, including the measuring range, hardware filtering bandwidth, and sampling rate. So once all these features are known, the duration of the pulse artifact is also known. And if the proper setting of the EEG amplifier are used, the duration of this artifact can be as short as a few milliseconds. So in our software, we can easily mask the pulse artifact by replacing it with a flat line. In this way, we can adjust the amplitude range of the signal, and we can see EEG activity. But remember, this artifact is only apparently gone, because it is still present in the raw data recorded by the proprietary software of each amplifier. So we have to remember as a first pre-processing step to replace this artifact with a smooth fake signal before doing anything else, such as filtering, independent component analysis, and whatever. Great, so let's restart our measurement session after masking the pulse artifact. We deliver a single pulse. What do we see on the screen? Well, at this stage, we might still see an artifact. This artifact is not as big as the pulse artifact, but is certainly larger than brain activity. It spreads almost everywhere, and it is characterized by a weird time course occurring in the first tenths of millisecond after the pulse. It is the recharge artifact. This artifact is produced by the fact that the stimulator's capacitors need to be recharged between subsequent pulses. So in case there is a transient current flow in the coil or a change in the potential of the coil during the recharging, this artifact appears in the EEG. Since the stimulator's capacitors usually recharge immediately after pulse delivery by default, we can easily remove this artifact by delaying via software the time of recharging beyond the temporal window of interest for TMS evoked potentials. Great, so we can restart our measurement session after masking the pulse artifact and delaying the recharge artifact. We deliver a single pulse, and what do we see now on the screen? Well, the signal is much better than in the previous examples, but still is contaminated by an artifact. This artifact is small compared both to the pulse artifact and to the recharge artifact. But it's quite annoying for two reasons. First, it occurs immediately after the pulse, but then decays for several tens of milliseconds. Second, it is more likely to find this artifact in the EEG channels nearby the stimulated site. It is the discharge artifact. The discharge artifact is produced by the fact that TMS pulses may charge all the capacitances located along any possible induction current path, such as at the electrode gel interface and at the gel skin interface. Immediately after being charged by the pulse, these capacitances discharge and produce a decaying artifact. So in order to reduce this artifact, it is important to carefully prepare the skin for lowering its resistance and to use EEG electrodes with a shape that facilitates skin preparation. Great, so let's restart our measurement session. We did everything well, let's assume this. So we deliver a single pulse, and I want to highlight here that at this point it is very important to be able to visually inspect single trial EEG responses to TMS in real time. Why is this so important? Because if we are blind at this stage, we can deliver many pulses, we can collect hundreds of trials, then the experiment is over, and the day after, when we analyze our data, we may find something like this: average TMS evoked potentials severely contaminated by large muscle artifacts. Well, at this point, it is too late. We cannot do much with our data. But conversely, if we were able to visually inspect single trial EEG responses to TMS in real time, we could immediately recognize the presence of muscle artifacts superimposed to EEG activity after a single pulse. Indeed, muscle artifacts show very peculiar characteristics that make them easily recognizable on the signal. First, they can be quite large in amplitude. Second, they are usually characterized by a biphasic shape, followed by slow return to baseline. Third, they are largest over the lateral aspects of the head. Muscle artifacts are produced by either direct activation of muscular fibers by TMS or by activation of the nerves innervating the muscles. Muscle artifacts are particularly annoying for two reasons. They, for sure, are superimposed to genuine EEG activity. But most importantly, skull muscle twitches can be perceived by the examined subject, and thus can potentially produce unspecific brain responses to sensory stimulation. That's why it's very important to maximally prevent the occurrence of muscle artifacts by adjusting TMS parameters. This task is not as difficult as it could seem at first glance. I will show you an example. Let's zoom on the lateral channels where muscle artifact is larger. This is real data, and I can tell you that in this case, if I stimulate the same subject in the same cortical spot at the same TMS intensity by slightly rotating the coil by 30 degrees, I obtain this other single trial EEG response, which is devoid of muscle artifacts. Now, this is just an example, I'm aware of it, but in my experience, very often a slight change in the coil orientation or a tiny shift in the coil position are enough to successfully reduce muscle artifacts without significantly changing the overall stimulation parameters. Okay, so let's focus on this single trial response. Let's zoom out, and this is a single trial EEG response to TMS devoid of large visible artifacts. So we can keep these stimulation parameters, location, intensity, orientation of the induced field, and we can start delivering trials, because at this point we want to see how average TMS-evoked potentials, TEPs, look like. At this point, it is very important to be able to visually inspect average EEG responses to TMS in real time. Why is it so important? Because if we are blind at this stage, we can deliver many pulses, collect hundreds of trials, then the experiment is over, and the day after, when we analyze our data, we may find something like this. In this case, post-stimulus EEG responses to TMS are not significantly different from baseline activity. So, unless we were delivering TMS stimuli over a cortical lesion, which is unlikely to be the case, this result can be explained only by saying that TMS in this experiment was not effectively stimulating the cortex. But if we are able to visually inspect, let's say, the first 20 trials average of EEG responses to TMS, we can early observe whether this situation is occurring. So we can stop recording, adjust TMS parameters, and increase the impact of TMS on the cortex in order to obtain a measurable signal. But how should we look in real time at average TMS-evoked potentials? Well, let's make a step backward. When we say potentials, we actually mean potential differences between single channels and a common physical reference site. So skull potentials are always affected by the location of the physical reference, because in our body there are no neutral reference sites. It is clear if we look at TEPs in common reference. As you can see from this example, the waveform of TEPs across channels is very much correlated, and this is an effect of having a common physical reference site. To solve this issue, we can re-reference each channel to a linear combination of the recordings from all channels, and this solution is called average reference. But when computing average reference, we have to remember to exclude from the computation of the average reference bad channels such as this one, because otherwise, after re-referencing, this artifact is spread everywhere. In addition, when we compute the average reference, we have to exclude flat channels such as this one, because otherwise, after re-referencing, this signal does not appear as flat anymore, but this is clearly an artifact. So the correct solution is to compute TEPs in average reference using good channels only. In this way, we can really observe the characteristics of EEG responses to TMS. And in this example, I believe that the oscillations that you can see here, right after the pulse, are genuine because they show the typical characteristics that are expected from brain responses to direct stimulation. In particular, you can see that the largest evoked oscillations are located nearby the stimulated site, as compared to distant channels. Second, the largest EEG responses appear immediately after pulse delivery. Third, there is a left to right asymmetry in amplitude that is in line with the stimulated hemisphere. In addition, genuine EEG responses to TMS should also show the following characteristics. If I stimulate even at the same intensity two different cortical spots, I should obtain different EEG responses to TMS, with the largest responses that follow the stimulation site. Stimulate exactly the same cortical spot at different intensity, I should find different EEG responses to TMS with larger responses to stronger stimulation and weaker responses to weaker stimulation. Stimulate exactly the same cortical spot at the same intensity but with a different orientation of the coil, I should obtain different EEG responses to TMS. All these examples I just showed you have been obtained by averaging the first 20 trials only. Here I show you that a visual inspection of the average obtained by the first 20 trials of a longer session is already enough to have a reliable idea of the amplitude and latency of the main EEG responses to TMS. Because as shown in this picture, the latency and amplitude of EEG responses to TMS is coarsely similar when comparing the 20 trials average with a 100 trials average. Well, I'm going now towards the conclusion of my talk, and so far I've shown how to recognize the characteristics of genuine EEG responses to TMS in real time during an experiment, already while you are performing your measurement. But a more rigorous and quantitative demonstration that genuine EEG responses to TMS are sensitive to perturbation parameters and repeatable over time actually dates back more than 10 years ago. And again, I want to highlight that spurious components potentially elicited by the sensory stimulation associated with TMS have very different characteristics as compared to genuine EEG responses to TMS, such as these ones. Here you can see EEG responses to TMS applied over the left premotor cortex in red and applied over the left motor cortex in blue. As you can see here, the two TMS evoked potentials are rather different. They have different time course and different instantaneous scalp topographies. Conversely, EEG responses to sham stimulation performed to mimic the sensory stimulation possibly associated with TMS typically show simple time courses and an instantaneous scalp topography that does not differ when sham stimulation is applied on different scalp sites. I can show you here some examples. These are the EEG responses to TMS. EEG responses to sham stimulation performed by delivering either auditory stimuli or scalp electrical stimuli, specifically designed to mimic, respectively, the coil click and vibration. As you can see here, EEG responses to sham stimulation are characterized by simple waveform, and instantaneous scalp topographies are rather comparable and very similar, even when sham stimulation is applied over different scalp sites. Last, even when the intensity of scalp electrical stimulation is increased until the subject complains, clearly EEG responses become larger than before, but they are always simple and with a stable topography. So to conclude, we can say that artifacts and spurious components usually have a weird or a stereotypical waveform. Conversely, genuine TMS-evoked EEG responses are site-specific as well as intensity and orientation dependent. And this sensitivity to stimulation parameters is not limited to the first milliseconds after pulse delivery, but is present also at middle and long latency for several hundreds of milliseconds after the pulse. My take-home message here is that in order to collect genuine EEG responses to TMS, it is very important to be able to inspect in real time, visually inspect in real time, the EEG responses to TMS. Because only in this way, with this possibility, we can empirically adjust TMS parameters to minimize the contribution of artifacts and maximize the impact of TMS on the cortex at the same time. Thank you very much for your attention, and I'm open for questions.
Okay, thank you very much, Dr. Casarotto. We are now going to open the Q&A session. As a reminder,